Hello guys, welcome to Buddy Academy. This is Uma here and I'm going to assist you in biology. So before we start our lesson, let us talk about this year which was full of slides. So people around the world slid in many things, right? So where did you students slid? You slid in your studies and your exam preparation. So yes, are you worried to face your board exam now? Don't worry because I'm here. Stay cool and I'll be giving you some concepts and questions on biology based on which you can face your exam so confidently. So before going into our class, I will also tell you something that I will provide you some tips in each class and you can apply them and you can tackle your exams easily. So here we go. Tip number 1. The first tip is to defeat yourself. So you should always remember that there is no strong competitor for you than yourself. So first thing is you have to compete within you and defeat yourself. How to defeat yourself? Eventually the question comes. Yes, you have to break all the barriers and all the negativities that you have. So first thing is break the barriers. Never think you cannot do anything. Always start with a positive note. I can do and I will do. And where can you implement this? In your exams. So how can you defeat your previous exams? By having a better score than your previous exams. So what you have to do for that? You have to start analyzing your papers and you have to analyze and mark the areas of your weakness, work on that and at least try to score 10 to 15 marks higher than that, so that you will be able to get a fruitful result at the end of the year. So let us move on to our chapter now, and today we are going to discuss about the second chapter, that is sexual reproduction in flowering plants. So you know this falls under the unit reproduction. So you very well know that this reproduction unit has 4 chapters, right? So why we are starting with the second chapter? Because this year due to pandemic, some syllabus has been deleted by the CBSE. So if you are not aware of that, immediately go to cbse.nic.in and check out all the deleted syllabus for all the lessons, that is for all the subjects, so that before starting your preparation you will be aware of the current syllabus. And you should also remember that this is only for the current academic year and only for your board exams and not applicable to your school exams. So yes, we are going to start with the second chapter under the first unit. So the weightage of your first unit will be 14 marks, where you will have 3 chapters now, that is sexual reproduction in flowering plants, human reproduction and reproductive health. You all very well know that this reproductive health chapter is a small chapter and it only has weightage of around 4 marks. So you can plan now. So this sexual reproduction in flowering plants and human reproduction will have a weightage of around 10 marks. So you should not even omit a single chapter because already chapters has been reduced. So be thorough with each and every concept in this chapter. So in this chapter, specifically sexual reproduction in flowering plants, what are we going to deal about? So first let me start with flowers. So what are flowers? So how are they? So they look colorful, they are centered, they are very bright, right? So what for they are like this? For us to see and admire? So no, it is for their own purpose. And for what purpose? That is what we are going to deal with this chapter, because they have to do reproduction. For reproduction, pollination has to happen. So some pre-fertilization and post-fertilization events will also be present, which we will be discussing in this chapter. So without any delay, come on, let's go to our class. So our first topic will be flower, a fascinating organ of Angiosperms. So this can come for 3 marks, but it is not very important, but then you have to know this. So a flower basically consists of calyx, that is sepal, corona petal and androecium and a gynoecium. So you know the stamen or androecium is the male part of the flower and it contains anther and filament. Filament will be long, slender like stalk, and then pistil or gynoecium, you can also call that as gynoecium, and it contains 3 major parts: stigma, style and ovary. So you may get this as a 3 mark for drawing and labeling, but very rare. So for just knowing purpose I am explaining this. So next will be pre-fertilization structures and events. So events are structures which are happening before fertilization. In that, first we will be discussing about the male part, that is stamen and microsporangium and a pollen grain. So parts of a typical stamen. Stamen consists of a long style called the filament and the terminal end is generally bilobed. So you can see the terminal end is bilobed structure containing an anther. So it is an bilobe structure and the proximal end will be attached either to the petal or the thalamus of the flower. So you can see this is an enlarged view of an anther. The proximal end is attached to the petal of the flower. The number and length of the stamens are variable in different flowers. So again this can come for 1 or 2 marks. So all the stamens in the flower, in every flower are not same. Even numbers are not same, they may vary in count and size. So a typical angiosperm anther is bilobed. Each lobe will be having 2 theca. You can see here it is bilobed, but each lobe is having 2 thecas here, and a longitudinal groove will be running by separating these 2 theca. So an anther is typically a 4-sided structure consisting of 4 microsporangia located at the corners, that is 2 in each lobe. So these microsporangia develop further and become pollen sacs and extend longitudinally through the length of an anther and are packed with pollen grains. So again I will explain this. This is nothing but a structure of an anther. Again they are tetragonal consisting of spore microsporangia, that is 2 are present in each lobe. So structure of microsporangium, again I have given a star mark which is indicating you it is a very important 5-mark question. So in a TS of a typical microsporangium, so we saw that it will appear nearly circular, but it is not circular. So it is generally surrounded by 4 walls like epidermis, endothecium, middle layers and tapetum. So all the outer walls perform the function of protection and help in dehiscence of the anther. So what are the walls? While showing you the picture, I will be telling you again. So what are the walls that are present? Epidermis is present, endothecium and middle layer. So this middle layer you should note because it is not a single layer, it is like 3 to 4 layers, and these will help in protection and dehiscence of the anther. So outer walls perform the function of protection and dehiscence of anther to release the pollen. The innermost wall will be tapetum. It nourishes the developing pollen grains, and tapetum will have a dense cytoplasm and they will also have more than 1 nucleus. So we have seen there are 4 layers present. What are they? First one is epidermis and the second one is endothecium, third one is middle layer and the fourth one is tapetum. So this is a typical anther structure. So it is like nearly circular, but it is tetragonal. So I am not mentioning all the layers here. Let us consider this as the epidermis and this as the endothecium. And again I will give you one more circle which will indicate middle layer, and inside middle layer I will just draw the tapetal cells. So again this will have something called sporogenous tissue. So this sporogenous tissue will mature and get converted into pollen mother cell, that is PMC. So in each lobe all these events will happen. I am mentioning this for better understanding. So this PMC will divide meiotically, that is reduction division, to give pollen tetrads. So they will be present as 4. How will be pollen grains present? They will be present as an aggregate of 4 and they are also called as microspore tetrads. So here these are called as microspores and the female gamete is called as megaspore. And these microspores, let me write tetrad alone here. So again what will happen? These will dehydrate and form individual pollen grains. They mature, dehydrate, that is lose water, and they form individual pollen grains, and during the time of dehiscence they are released from the anther. So when the anther is young, a group of compactly arranged homogeneous tissue called sporogenous tissue will will occupy the center of each microsporangium. So in all the center this will be present. So again I am mentioning this for better understanding. So what is microsporogenesis? As the anther develops, the cells of sporogenous tissue will undergo meiotic division. Here you have to see this will undergo meiotic division, that is reduction division. Let me just put a minus here so that you will be understanding. Meiotic is nothing but reduction division, where the reduction in number of chromosomes will occur. So as each cell of sporogenous tissue is capable of giving rise to a microspore tetrad, it is called as a pollen mother cell. So initially it is called as a sporogenous tissue, then it is called after maturation, that is after growth, it is called as a pollen mother cell. What this pollen mother cell will do? This will give rise to microspore tetrad and they will be dehydrating and forming pollen grains. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. So again, this is the structure of microsporangia, which will come for diagram purposes, and you have to practice this diagram. So you can either draw like this, or you can just put up 4 structure like tetragonal structure, and you can also mention all these layers inside that, but that will be time consuming. This will be an easy process for you to draw. You can easily draw this middle layer. Connective is nothing but an infertile tissue that is present in the middle part. So these are the middle layers. You can see the first layer is the epidermis, second layers is the endothecium, and as I said, middle layers are 3 to 4 layered cells, and then you also have the tapetum. So this tapetum will have a dense cytoplasm and more than 1 nucleus, and inside that only microspore mother cells are present. So you can see this is the dehiscence of anther, and from that pollen grain, these are the pollen grains which will be released. So these are the different structures of tetrad. So here this A structure, this first structure is only very important because microspore tetrad generally form in this type, that is 1, 2, 3 will be facing front side, whereas the fourth one will be at the back side. So this is generally called as tetrahedral, and most of the pollen grains will form this tetrahedral structure. So next one is isobilateral. Here all 4 are seen from the front view. So again decussate is nothing but one more thing will be, that is one more pollen grain will be arranged backwards, and next will be T shape, and the last one will be linear one, where everything is arranged in a linear fashion. So as the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of the anther. So after microspores are formed, now we have to learn the structure of a particular pollen grain. So now what is happening? So this is the process of microsporogenesis. So here you can see the microspore tetrad and the released microspore, and what is happening here. So it is undergoing mitosis. After this only mitosis is happening and no meiosis is happening. So you should definitely note that only till microsporogenesis, meiosis, that is reduction division, is happening. After microsporogenesis, that is release of microspore, there will not be reduction division, there will be mitosis. So mitosis 1 and after mitosis 2, what will happen? So after mitosis 1, what generally happens is the pollen grain will have 2 nucleus. One is the vegetative nucleus and the other one is generative nucleus. So this generative nucleus will be present inside a generative cell. You can see here, this is the structure of a pollen grain which can come for 2 mark or 3 mark. So when you do your practical exam, this is an compulsory question for everyone and you can expect this for 100%. That is structure of pollen grain and germination of pollen grain will definitely come. So this pollen grain will have a hard outer covering called exine, and this exine will contain something which is called as sporopollenin. So this is a substance, sporopollenin. It is a chemical substance which is very hard substance. This exine is very hard because of the presence of this sporopollenin. And one question, why this exine should be hard? You may think that the answer is pollen grain should travel several miles apart and then they have to fertilize. So, to maintain their structural integrity, this exine is very hard. So, due to this presence of sporopollenin, pollen grains are also stored or also preserved as fossils. Then they have intine, and this germ pore is present here where the pollen tube will be developing, which we will be discussing later. And you can see a small cell that is floating inside this cytoplasm of this large cell, and that is the generative cell, and this is the generative nucleus, and one more nucleus that is present is the vegetative nucleus. You have one speciality in pollen grain. What happens? Sometimes pollen grains are released in this condition and they are called as two-celled condition or two-celled or even two-nucleated condition. So, this is one more very important thing to remember sometimes. So, you can see after pollen mitosis 2, you can see this tricellular pollen, right. So, this is 3-nucleated. The generative nucleus will divide and it will form a 3-nucleated conditions. You should remember most of the pollen grains 60% are shed in this 3-nucleated condition, but others are shed in this 2-nucleated condition. So, during the development of pollen tube, again this mitosis 2 will happen and they will just transform into 3-nucleated conditions, and this will divide to form the male gametes of the flower. So, yes, pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. As I have already mentioned, sporopollenin is present, and then why it is that important? Because it can withstand high temperature, strong acids or alkali, and till now no enzyme has been found in this earth to degrade this sporopollenin. So, pollen grain hexine has prominent aperture called germ pores where this sporopollenin is absent. So, we have seen the germ pore. So, here this sporopollenin component is absent. Why? Because they have to develop the pollen tube only from this place. So, yes. Why do you think hexine should be hard? So, I have again given those answer to you, because pollen grains has to travel to several places under several conditions and then it has to undergo fertilization. So, the inner wall of the pollen grain is generally called as intine, and it is a thin and continuous layer which is made up of cellulose and pectin. Again, the cytoplasm is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells. As I said, one is vegetative and the generative cell. Vegetative cell is bigger, whereas generative cell is small and it will float in the cytoplasm of vegetative cell. In over 60%, pollen grains are shed at this two cell stage. So, mostly in most plants it is shed in two-celled stage. In remaining species, the generative cells divides mitotically to give rise to two male gametes before the pollen grains are shed, and they are called as 3-cell stage or 3-nucleated stage, even you can tell them. So, you should remember why it is called as 2-celled and why it is called as 3-celled, because these 2 points are very important to remember. So, is pollen grain having any hazardous damage to your health? Yes, it can cause asthma, bronchitis, etc. So, majorly where did this pollen allergy come from? So, it came from carrot grass that came as a weed with wheat, that is with imported wheat. It came as a weed, that is undesirable plant, and now it has become ubiquitous, that is it is occurring more and causes pollen allergy. So, you should remember carrot grass which causes pollen allergy. How did it come? It came with imported wheat as a weed. So, then it can cause respiratory disorders like asthma and bronchitis. So, how viable are pollen grains? One more important one mark is this. So, how can we store pollen grain? It can be even asked as a 2 or 3 mark question. So, some pollen grains are highly viable. So, in some cereals like wheat, pollen grains will lose the viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, Solanaceae, and they can even maintain viability for months. So this pollen viability is highly variable from species to species. You must just remember, some cereals like rice and wheat will lose viability within 30 minutes, whereas some other plants can even maintain this viability for many months. So you may have heard that semen storing like sperms of humans or animals are stored for artificial insemination. Likewise, pollen grains are also stored in pollen banks in liquid nitrogen in -196 degrees Celsius. So like in pollen banks, so these can be used as reserve for pollen grains. So again, liquid nitrogen -196 degrees Celsius. So do these have any other advantages? Can pollen grains be used in any other ways? Yes, they can be taken as supplements. So what you see here is pollen syrups and pollen tablet. So in Western countries, these are used for a larger extent, and what is the main use of this? These are shown to increase the performance of athletes and race horses. So again, it is taken as food supplements. Who is using that? Majorly in Western countries, and who are mainly benefited by this? It is increasing the performance of athletes and race horses. So the next topic will be pistil, megasporangium, that is ovule, and an embryo sac. A flower can be divided into two types based on the number of carpels. Carpels are nothing but again pistil, female part of the flower that is present. Monocarpellary and multicarpellary. Mono, as the name indicates, it only has one carpel, whereas multicarpellary has many carpels. So this multicarpellary is again divided into two types, that is syncarpus and apocarpus. Syncarpus have united carpels and apocarpus have separate carpels. So you can see this is the structure of a pistil which contains stigma, style, and the ovary. Inside the ovary there will be ovules which is attached to the ovary by the placenta. So here stigma will serve as the landing platform for the pollen grains. All the pollen grain will come and land on the stigma. So examples of multicarpellary syncarpus pistil is papaver, and multicarpellary apocarpus pistil is michaelia. So again, there can be one or many ovules that can be present in a particular ovary. For example, when you take mango, you can only see one seed there. So you can conclude that its ovary has only one ovule, whereas some other fruits will have many ovules. There it can be many ovules also. So an ovary can contain one to many ovules. So the stigma serves as a landing platform for pollen grains. The style is elongated slender path beneath the stigma. The basal bulge part is the ovary. Inside the ovary is the ovarian cavity where the placenta is located. Arising from the placenta are the megasporangia, commonly called as ovules. The number of ovules in an ovary may be 1 to many. So this is the structure of a megasporangium, that is an ovule, which can be asked for 5-mark question, and this is a very important concept that you should learn. Ovule will consist of an hilum. The initial part is generally called as the funicle. So funicle will be attached to a hilum. So let me draw a funicle first, and this funicle will be attached to an hilum, and then this funicle is the place, or it is the region where this placenta is being attached to the ovule, and the body of the ovule fuses with the funicle in the region called hilum. So let me name this as funicle and the region as hilum. So this is hilum. Again, it has two protective coverings. This ovule has, though they are called as integuments. The outer is called as the outer integument, whereas the inner one is called as the inner integument. So as I said, two protective coverings are present. The outer one is the outer integument, and the inner one will be the inner integument. So this integument will cover the entire ovule, leaving a small space. This is called as the micropylar end, and this closed end is representing the chalazal end. So it is also called as the base of the ovule. But during fertilization, you should remember that this chalazal end will generally be upside. Only then it will be easy for us to understand the process of fertilization during the development of a pollen tube. So inside these integuments, what is present? Nucellus is present. Nucellus are mass of cells which provide the nutrients for the developing embryo. So you can see here, this is the developing embryo and these are the cells of nucellus. And what are these protective layers called as? They are called as the integuments. So this is the outer one and the inner one will be the inner integument. I will just write the integument here. So clear? Yes. Enclosed within integument is a mass of cells called nucellus. Cells of nucellus will have abundant food material. Located in the nucellus is the embryo sac or the female gametophyte. So which is going to produce the female gamete. So yes, an ovule generally has a single embryo sac from a megaspore through reduction division. In majority of flowering plant, one megaspore is functional while other three degenerate. Only the functional megaspore develop into female gametophyte or the embryo sac. So this method of development is generally called as monosporic development. You can see from here, this embryo sac, this megaspore mother cell, that is MMC, will divide by reduction division, again meiosis, into 4 cells. You can see here this megaspore dyad, this is MMC, that is megaspore mother cell, also called as MMC, and this is dividing into 2 by meiosis 1 and again dividing into 4 by meiosis 2 and then forming megaspore tetrad similar to sperms. But here what happens, out of these 4 only one is functional and from this one the entire embryo sac is developing and hence it is called as a monosporic development. So after this all the reduction division stop. So after megaspore tetrad, for example this is the megaspore tetrad, let me take all these 3 things are degenerating. Let me strike out all these 3. From here onwards what will start, it is going to be mitosis and not the meiosis. Clear? So yes, you can see again this will again divide by mitosis 2 to 4 and again 4 to 8 and 4 will be arranged at each pole, that is micropylar end and the chalazal end. You can easily remember these structures. So you just think as 4 are segregated on each side. From each side again each one is coming to the central and it is called as the central cell. So here you should note that after immediate nuclear division there is no cytoplasm formation, which means that the number of nucleus will not be equal to the number of cells present. So this large central cell will contain 2 nucleus. So this will be the chalazal end. Let me again tell you. So when we are learning about fertilization, chalaza is the basal part, but then it should be kept at the top, only then we can understand. You can see that at chalazal end there will be 3 antipodals. You can easily remember this. A chartered accountant is going to an FM station. You can easily remember it like that. Chalazal end will contain 3 nucleus and 3 cells which are called as antipodals, and the micropylar end, so that is what I have given M here, will contain filiform apparatus. Filiform apparatus is nothing but an apparatus which is going to guide the pollen tube into this particular cell, and then it will have 3 cells. So as we said, 3 are present at this end, right? So here what will happen, 2 will be synergids whereas 1 will be the egg cell. So 2 will be synergid and 1 will be the egg cell, and this is generally called as an egg apparatus. So if you can understand, a CA is going to FM station, just add one more E here so that it will be clear. At chalazal end antipodals are present, at the micropylar end filiform apparatus and the egg apparatus are present, and then what is happening to the large central cell? It is containing 2 polar nuclei, and hence the fertilization is called as double fertilization, because 2 male gametes that are present is going to fuse egg and the polar nuclei that is present. So it is called as double fertilization and triple fusion. So again, this is one more important 1 mark question. What are the ploidy of the cells of nucellus? It is 2n. Again, ploidy, you need not mention any number, just write n or 2n, 3n, 4n, whatever it is. So again, megaspore mother cell, it is 2n, functional megaspore n, female gametophyte n. Hope this would have been clear to you. So the nucleus of the functional megaspore will divide mitotically to form 2 nuclei, so which will move to opposite pore, then it will be forming an 8 nucleate cell of the embryo sac, and you should remember that it is strictly free nuclear. All nuclear divisions are not immediately followed by cytoplasm. As I said, out of these 6 are surrounded and organized into cells on 2 sides, that is antipodals and egg apparatus, whereas the 2 nucleus remaining will be in the center forming polar nuclei. So these synergids will have cellular thickenings at the micropylar end, which is called as filiform apparatus, as I said, which will guide the entry of the pollen tube. So the large central cell has 2 polar nuclei. Thus a typical angiosperm embryo sac is 8 nucleate and 7 celled, or you can even mention it as 7 celled and 8 nucleated. So this point is very important to note. A typical angiosperm embryo sac is 7 celled and 8 nucleated. They can also ask justification for that, and you should be able to explain all those antipodal story and then those egg apparatus. And since it is not strictly free nuclear, they are having 2 polar nuclei at the center and a large central cell is formed. So what next? We have seen that these things develop. So what process will happen next? So the male and female gametes should meet, right? So that is called as pollination. Transfer of pollen grains from the anther to the stigma is generally termed as pollination, but what the definition is given here, transfer of pollen grains to the stigma of a pistil is generally termed as pollination. So pollination is of 3 types depending upon the source of pollen, whether it is from the same plant or from the different plant. Sometimes same plant, different flowers can also be formed. So first one is autogamy, it is between the same flower, that is pollination will be happening between the same flower. Next one is geitonogamy. Geitonogamy, you have to note that it will happen between the same plant but different flower, same plant, different flower. Xenogamy, between 2 different plants. Again, autogamy is achieved within same flower, so because it is very closer to each other, but you should note that autogamy is very rare. Why? Because autogamy requires synchrony and stigma receptivity, that is development and maturation of both male and female gametes should happen at the same time, and the stigma should be developed to accept the pollen. So because of this thing, autogamy is very rare. The anthers and stigma should lie close to each other. One more condition is both anthers and stigma should be present very close to each other, only then autogamy will occur. Where complete autogamy can be seen? Some plants such as viola, that is common pansy, oxalis and comolina will produce 2 type of flowers. General flower is called as chiasmogamous flowers, which is nothing but open flower, which we see. So there are some flowers which are called as cleistogamous flowers. So importance of this cleistogamous flowers are very important 2 mark question. So it can even come in 1 mark question. Cleistogamous flowers are flowers which do not open at all. They do not open in their lifetime. So chiasmogamous and cleistogamous. Cleistogamous flowers are very important because they do not open in their lifetime. They remain closed all the time, like they remain in bud state throughout their life. So that what happens, male and female gametes are present very next to each other. What will happen? Eventually fertilization will happen. So but there is one advantage for this cleistogamous flower. So what is that advantage? These flowers can produce assured seed set even in the absence of pollinators. So we will be discussing pollination requires some pollinators. So these pollinators are required for transfer of these pollen grains, and again Cleistogamous flowers do not require them. Even if they are absent, they will produce seed set. So this is an viola plant. You can see the closed flower and open flower, the assured seed pod that is formed after autogamy. So geitonogamy, as we have seen, transfer of pollen grain from the anther of stigma to the other flower of the same plant. So even this is also similar to autogamy because it is happening between the same plant and there is no genetic variation. So xenogamy, again different plant will be present here. So here what happens, only in this process there are genetically new progenies present. So how pollen grains are transferred? They are transferred through some agents. What are they called? So self-pollination, let us leave, there is no agents required. Cross-pollination, xenogamy or autogamy, requires abiotic and biotic agents. So abiotic are wind and water, anemophily, hydrophily, and biotic agents are many insects, and out of these, bees are very important for pollination. So this is a very important remark which you should remember, that all the points, like you should be remembering each and every point, because this is a very important remark which is nothing but characters of insect pollinated flower, wind pollinated flower and water pollinated flower. What are the different characters? So insect pollinated flower is very important among them. The flower is large, colorful, scented and has nectar. So you can see why the flower is large and colorful. To attract all the insects. To attract all the insects, the flowers are very large and colorful. To give some return gifts to them, these flowers also has nectar. So whereas in wind pollinated flowers, they are small, not colored, no scent and no nectar, and even in water it is very small. So coming on to the type of pollen grains, not large in numbers, sticky or spiny. Why they should be sticky or spiny? Because when the insect comes and sits on that, it should stick onto the surface of the insect and the insect should carry. So whereas you can guess it for wind now, it will be large in numbers, it will be lightweight and it will be very dry, and one more thing, it will be loosely attached. So you can conclude from this, if something should be carried away by wind, what should it do? How it should be? It should be very lightweight, it should be dry, it should not be sticky, it should not go and stick to some other substance, and it should be loosely attached from that plant so that it can easily be released and carried away. So whereas in water, example Vallisneria, pollen grains will float till they meet the female flowers. Here in water, the pollen grains are generally long and ribbon like. So stigma here will be sticky and does not hang out of the flower. Here it will be feathery and hang out of the flower. Why it should be feathery here? Because only then it can trap the pollen grains that are present on the wind. Here it is dioecious. So sticky, again why it should be sticky and does not hang out of the flower? The insect should go inside and sit them and transfer the pollen grain. So this is an insect these are insect pollinated flower. You can see how colorful and vibrant they are, and this is the corn cob. You can see this is a wind pollinated flower. This is the stigma and how the pollens are hanging, that is the male part are hanging. You can see how exposed the anther is. So again this is the flower of the corn cob and stigma which is ready to trap the pollen grains, and this is Vallisneria which is going to trap which is an water pollinated flower. So next topic will be outbreeding devices. So what are outbreeding devices? Plants will have some mechanism to prevent self-pollination. So why they are preventing self-pollination? Because they have some depression. If they are continuously undergoing autogamy, they develop some depression which is called as inbreeding depression, and there is no genetic variation produced. So what are the techniques used by them? So as we saw in first thing where autogamy is rare, the same technique. Pollen release and stigma receptivity are not synchronized. They both do not happen at the same time. Anther and stigma are placed at a different position. Then third device is to prevent inbreeding is self-incompatibility. They will not accept the same pollen. Stigma will not accept the same pollen. And then one more thing is production of unisexual flowers. So here if unisexual plant of one sex is produced, flower of one sex is produced, what happens eventually? Self-pollination will be stopped. So again, if both male and female flowers are present on the same plant, that is monoecious, and in several species, like one plant will have only male flower whereas the other plant will have only female flower, that is dioecious. Monoecious is single plant will have flowers which will either be male or it will be female. Both male and female flowers will be present, whereas dioecious plants will only have, for example, if there is one papaya tree, it will be male. One more papaya tree, it may be male or female, like that. So next will be pollen pistil interaction. So pollination does not guarantee the transfer of right type of pollen. We have several type of pollen grains, right? So how this will guarantee the pollination, that is after pollination fertilization. Sometimes wrong pollen can also land on that stigma, right? We cannot tell when to carry the correct type of pollen and land on that particular stigma, right? So that because of that only the pistil has the ability to recognize the pollen whether it is of right type or not. So how can it recognize? It can either accept or it can reject. So how can it do this? If it is the right type, it will accept and promote the pollination, that is post-pollination development of pollen tube, which will lead to fertilization. If it is of wrong type, it will reject. So how this is doing? Are they talking to each other? And yes, there is a dialogue happening between them, and that dialogue is mediated by the chemical components. It is the chemical dialogue that is happening. So it is only in recent years many scientists are working on this to mediate these chemical dialogues that are happening in between them. So what is the next process? Next process is germination of pollen grain. As I said, from the germ pore, the pollen tube will develop. It will eventually travel through the style and reach the ovary. So you can see these are the microscopic structures of germinated pollen tubes. And what will happen after that? Pollen grain, it will be developing pollen tube and the nucleus will be traveling. If the pollen grain is shed in a two-celled condition, what will happen? After the development of pollen tube, that mitotic 2, that is mitosis 2 division will happen to form a 3-celled condition, and these two are called as the sperm nuclei, and the vegetative nucleus which we saw will be the tube nucleus. So you can see how it is developing from stigma and through style and then traveling till the ovary and reaching the ovule here. So how it will reach? Through the gap that is present in the micropylar end. So in some plants, pollen grain are shed in 2-cell, whereas the other it is shed in 3-cell condition. Pollen tube after reaching the ovary enters the ovule through micropyle and then enters one of the synergids through filiform apparatus. Again, now we are viewing it upside down. Again, this is pollen tube, filiform apparatus. How it is guiding? It is guiding the pollen tube inside, and the tube nucleus is the secondary nucleus, and these are the male gametes that are present. So yes, you can see this. Again the same thing, fusing of polar nuclei. Again with one male gamete, the tube nucleus will eventually degenerate and only these two male gametes are functional. Here one is going to fuse with the egg and the other is going to fuse with the polar nuclei. And again this diagram is also very important diagram. You should remember the diagram, how it is reaching, how it is moving through filiform apparatus and it is fusing with polar nuclei. So yes, filiform apparatus present at the micropylar part of the synergid guides the entry of the pollen tube. All these events from pollen deposition on the stigma until pollen tube enter the ovule are together termed as pollen pistil interaction. So what are they? Events from pollen deposition till what? Till the pollen tube enter the ovule are generally termed as pollen pistil interaction. So yes, then if this interaction happens in nature, then many hybrid varieties are being produced now, right? So how they are produced? How desired characters are being transformed? So it is by artificial hybridization. So this artificial hybridization is nothing but we will not allow the unwanted pollen grain to mate or to fuse with our female gamete or our female plant that is present in order to prevent some unwanted characters. So but how is this achieved? How desired pollens are made to fuse with the desired female gametes? Is it possible? Yes, of course, it is possible through emasculation and bagging technique. Emasculation is nothing but removal of anthers from bisexual or hermaphrodite flowers. You can see these are the emasculation technique. If anthers are removed, are the flowers safe? Again, there will be cross pollination, right? After emasculation, a suitable bag is used to cover the flower. So this is the bagging technique. So emasculation, removal of anther, and then bagging technique to prevent that particular female part of the flower to interact with other pollen. Bagging technique is done. It is not very tightly covered. It is loosely covered with like butter papers or very soft slender papers like this. So yes, the next process is a very important process. As I said, it is double fertilization. Why it is called as double fertilization? Because here the polar nuclei are present. One of the sperm cell will fuse with this, forming an endosperm, that is with 3 ends. So we have seen zygote. So one will fuse with egg forming zygote, which will have a ploidy of 2 ends, whereas the ploidy of endosperm is 3 ends. So this, since fertilization is happening twice, it is called as double fertilization, and this endosperm formation is also called as triple fusion, and this double fertilization is a characteristic feature of all the angiosperms, that is flowering plants. And what this endosperm is going to do, it is going to provide nutrients for the developing embryo. So yes, after entering one of the synergids, the pollen tube releases 2 male gametes into cytoplasm of synergid. One of the male gamete moves towards the egg and fuses with nucleus to form zygote, whereas other will fuse with the polar nuclei forming PEN. PEN is nothing but triploid primary endosperm nucleus. So this will again divide, and again this division, PEN division, is also free nuclear, that is immediate cell wall formation will not happen. This should be noted because sometimes in some cases this endosperms are not completely utilized by the developing embryo. So the central cell after triple fusion becomes the primary endosperm cell, that is PEC. PEN is the nucleus and the cell used to denote that is primary endosperm cells, which will provide the nourishing embryo with nutrients. So these are the post-fertilization structures. After fertilization, as we said, some structures will develop, right? So these are the post-fertilization structures. These are endosperm, embryo and the seed. So endosperm development precedes embryo development. Why? As I said, this endosperm will have reserve food material. So this is again one important phrase or dialogue to remember, one question to remember, because endosperm will have the reserve food material, so its development is more preceding the embryo development. So yes, in most common type, the endosperm undergoes successive nuclear division to give free nuclei. So you should note that the number of free nuclei formed before cellularization will vary greatly, so that many will remain as free nucleus. So this endosperm may be completely consumed or it may be persisting in mature seed. For example, completely consuming, it is pea groundnut, and in mature seed it is castor and coconut, and the coconut water which we are seeing is the free nuclear endosperm, and only the surrounding white kernel is the cellular endosperm. So you can see this water is free nuclear endosperm, this white kernel will be endosperm cell. So this is a castor bean, a dicot embryo. You can see this. So this is again not utilizing the complete endosperm, it is leaving some endosperm. So again, what is an embryo? It develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after some amount of endosperm is formed. Why? Because for the nutrient. So stages of development of embryo in dicots, again you can see this is an globular embryo and heart-shaped embryo and a mature embryo. Again you should remember these diagrams: zygote, first division, globular, heart-shaped, mature embryo, contains cotyledon, plumule and radicle, which is nothing but root tip and shoot tip. Again this is a suspensor cell which acts like a ladder to push the embryo towards the developing embryo towards the endosperm. So yes, structure of embryo, again this diagram is very important. So the development process is similar, but the structure of a monocot and dicot embryo will vary. So you can see this plumule is the shoot tip and radicle is the root tip, and hypocotyle is the region present in between, or the joining place that is present in between plumule and radicle. You can see two cotyledons present since this is a dicot embryo. So again root cap will also be present. Hypocotyle, you should remember, between plumule and radicle. You will also have epicotyle, which is nothing but something above plumule, that is emerging above the ground surface or above the plumule, is generally termed as epicotyle. So again this is a monocot embryo, it possesses only one cotyledon, it is generally called as scutellum. Again in dicot we use the term plumule and radicle, but in monocot embryo we generally use coleoptile and coleorhiza. So we use this, like for example the plumule is covered with a sheath of cells called coleoptile, again the radicle is covered by a sheath of cells called coleorhiza, and epiblast is nothing but act as the second cotyledon for the monocot embryo. So again this is the structure of seed, seed coat, foot store and embryo. So you can see epicotyle, you can see some rudimentary leaves, and you know the layers of seed coat. The outer layer is called testa, inner is tegment, which you have already studied in your class 11. So again epicotyle, hypocotyle, radicle, seed coat, cotyledon. So this is a bean seed, dicot seed, two cotyledon will be present. So epicotyle above, hypocotyle, connection between cotyledon and the radicle. So seed is the final product of sexual reproduction, and it is often described as a fertilized ovule. Seeds are formed inside the fruits. They consist of seed coats, cotyledon and an embryonal axis. So generally mature seeds are of two types. One is non-albuminous and the other one is albuminous. As we already saw, the non-albuminous, the endosperm will be completely consumed. In albuminous, the endosperms will not be completely consumed. Again the same example can be cited here, pea and groundnut. Again here castor, wheat, maize, barley and sunflower. So this is one more important thing, and have tabulated that part of the flower and what it is going to become in a fruit or a seed. So you know that ovary will change into a fruit and the ovule will develop into a seed. So wall of ovary will change as wall of fruit, and the remnant of nucellus sometimes, very rarely, is present as perisperm. The integument will come as a seed coat. Wall of ovary, fruit coat, integument, seed coat. Just remember that. Then the micropyle will be present as a small pore in the seed coat. Why? Seeds generally dehydrate and they have a period of dormancy where they remain inactive. So during that time of dormancy, you know that seeds are also stored in larger amount for many purposes like agriculture and food storage. So in that time they will undergo dormancy and they will be dehydrated, they will be losing some water. So again when they have to regenerate, this small pore that is present on the seed coat will allow the entry of water and oxygen. So as ovules mature into seeds, the ovary develop into fruit. That is, the transformation of ovules into seeds and ovary into fruit will proceed only simultaneously. One more important thing is, fruits may be fleshy as it is seen in guava and orange, or it may be dry as it is seen in groundnut and mustard. So is there any relationship between number of ovules in an ovary and number of seeds present in a fruit? Definitely yes. If the fertilization is complete, the ovule and the seed ratio will be 1:1. So there are different types of fruits which are nothing but true fruit, false fruit and parthenocarpic fruit, which is very important. Parthenocarpic fruit, I will just explain it in the beginning itself. Development of fruits without fertilization. You should remember, if you have read the first lesson, that is reproduction in organism, parthenogenesis will be present where an entire organism will develop from the egg itself. Here also same thing is happening, development of fruits without fertilization. They are seedless fruits since they are not fertilized. So seed is a fertilized ovule. So since they are not fertilized, they are seedless fruits. Example, banana. So they are also used to produce many fruits like seedless watermelon, seedless papayas, like that. So true fruit is nothing but when ovary is developing into a fruit. False fruit is nothing but apart from ovary, if any part is developing as a fruit, that is a false fruit. Example, thalamus in apple, perianth in jackfruit. So you can see structure of a fruit. The pericarp contain outer layer exocarp, middle layer mesocarp and inner layer endocarp, and seed is present. Again seed will have seed coat. So these are the examples of false fruit, apple and strawberry, where thalamus is developing into the fruit instead of ovary. So what are the advantages of a seed? So seeds are products of sexual reproduction, so they have a better adaptive strategy, and as I said, they are capable of doing photosynthesis on their own, and they are also having new genetic combinations. So the next main thing is agriculture. So it is the basis of our agriculture. So as I said, dehydrating and dormancy property of that particular seed is crucial and essential for storage of seeds for further agricultural purposes or for the process of food. So how long do the seeds remain alive after they are dispersed? So same thing like pollen grains, they are also highly variable. So even for large number of years, sometimes several thousand years, they will be alive. So the oldest seed of lupine, Lupinus articus, is around 10,000 years old. It will have a dormancy period of 10,000 years, and recently 2,000-year-old seed of date palm was also discovered near King Herod's palace near the Dead Sea. So just remember these terms, that are enough. So it can vary. So highest dormancy period seed was found to be Lupinus articus. So next, our final topic will be apomixis and polyembryony. This is our final topic, but then as I said, each and every concept is very important. So species of Asteraceae and grasses produce seeds without fertilization called apomixis. We saw fruits without fertilization, but here seeds without fertilization. Apomixis is a form of asexual reproduction that mimics the sexual reproduction. So what is going to be done here? In some species, the diploid egg cell is formed without reduction division and develops into embryo and without fertilization. So diploid egg cell will develop into embryo without fertilization. So this is happening more often. This is seen in citrus and mango. So you should remember citrus varieties because questions are being asked from this part. More often citrus and mango varieties, some of the nucellar cells surrounding the embryo sac will start dividing and protrude into the embryo sac and develop into the embryos. In such species, ovule will contain many embryos and that is called as polyembryony. So what is going to happen in citrus and mango varieties? The nucellar cells that are surrounding the embryo sac will protrude inside and they will develop into embryos, and here what will happen? More than one embryo will be present, and that is referred to as polyembryony. So is this useful? Yes, because during the production of many hybrid varieties, more amount of cost, and apart from that, due to segregation of characters, we cannot able to get the particular hybrid and it is very expensive for farmers. So if they are recognizing these genes for apomixis, they can able to mimic that particular character and there will be no segregation in hybrid and they can produce it further, and scientists are also working on this particular area. So now we are going to discuss the previous year's question paper. This contains 5 years question paper where many questions are asked. So self-pollination is fully ensured if, what will be the answer? Self-pollination is fully ensured if the flower is bisexual. Even if the flower is bisexual, the maturation should be at the same time. So we cannot give this a tick mark. The style is longer than the filament? No, the flower is cleistogamous, and yes, this will be our answer. So what are cleistogamous flowers? Cleistogamous flowers are the closed ones, if you could remember. Okay, so draw a schematic transverse section of a mature anther of an angiosperm, and you are going to label epidermis, middle layer, tapetum, sporogenous tissue, and the connective. The middle part connective, please don't forget. A pollen grain in angiosperm at the time of dehiscence from an anther could be 2-celled or 3-celled. Explain. Again, as I said, during the release, sometimes the generative nucleus will not be divided. That is, it will not undergo mitosis 2. Sometimes it will be undergoing mitosis 2 and it would have divided. How are the cells placed within the pollen grain when it is shed at a 2-celled stage? Again, you have to draw a pollen grain here and you have to show when they are shed at 2-celled stage. So you are going to just draw a pollen grain and you are going to just show that at 2-celled stage there is going to be a generative nucleus, only one generative nucleus inside a generative cell and one vegetative nucleus. This is V vegetative and this is the generative. So differentiate between wind pollinated and insect pollinated flowers. As we have already discussed, this is a very important question. Insect pollinated will be, flowers will be large, it will be centered, it will be producing nectars, pollen grains will be sticky, it will be spiny, it can stick to surfaces of the insect, and the ovaries will be inside, it will not be exposed. Whereas wind pollinated, the female part should be exposed. The pollen grain, since it should fly, it should be very dry and it should be lightweight. Again, pollen banks are playing a very important role in promoting plant breeding program the world over. How are pollens preserved in pollen bank? At minus 196 degree in liquid nitrogen. How are such banks benefiting our farmers? So you should be thinking out of the box and writing these answers. So how these are benefiting? Much better pollen source and much better plant varieties pollens can be stored and it can be given to our farmers to produce better crops. So differentiate between parthenocarpy and parthenogenesis. This won't come because the first lesson will contain this topic and it is not present, but just then you can remember parthenocarpy and parthenogenesis. Parthenocarpy producing fruits without fertilization, that is seedless fruits. So this question is very important. Can a plant flowering in Mumbai be pollinated by pollen grains of the same plant species growing in New Delhi? Provide explanations to your answer. Since you are seeing bi-pollen grains, just don't imagine it should only be self-pollination. It can also be cross-pollination, right? We can collect the pollen, do emasculation and bagging techniques here. You should write it. Draw the diagram of a pistil where pollination has successfully occurred and label its part involved in reaching the male gametes to desired destination. Again, development of pollen tube, pollen pistil interaction diagram should be drawn here. So describe any devices in flowering plant which prevent both autogamy and geitonogamy. So what will prevent both autogamy and geitonogamy? That is self-fertilization, emasculation and bagging technique. Explain the events up to double fertilization after the pollen tube enters one of the synergid in an ovule of an angiosperm. So if it is a 5-mark question, it is must for you to draw the diagram, like which diagram we showed. It contains the pollen tube and then filiform apparatus guiding and all those things you have to draw and you have to explain. So this question is again very important. When a seed of an orange is squeezed, many embryos instead of one are observed. Explain how it is possible. So again, apomixis and polyembryony. If polyembryony is present, are they genetically similar or different? Comment. What is apomixis? Apomixis is mimicking of asexual reproduction in the sexual reproduction plants, right? So if there is asexual reproduction, what will happen? There is no genetic variation. So students, you have to think out of the box in order to answer many of the questions. This only comes when you continuously practice this. So with this, we are closing this chapter. I hope all the concepts would have been understood by you. You should go through all these concepts continuously and thoroughly in order to face your exams very confidently and have a clarity of all the concepts, and then only you can know how to answer a particular question. So what did we discuss today? We discussed about flower, events before and after fertilization. We have also discussed about structures that are formed after fertilization. So that's all for this class. Hope you have understood this class. So in next class, we will be discussing about two important chapters, human reproduction and reproductive health. See you students. I hope this class would have been very useful for you, and before going, it's time for you to start preparing. Schedule, prepare and then score. Thank you students. See you in next class.
For more queries, please contact Buddy on 98408 44987 or visit buddy.life.